to PowerPoint. And a while ago, I just emailed, okay, the PowerPoint for the last module. So we might go over some of them pag maaga yung natapos natin dito. And I still have another PowerPoint for the fluorescence. So this is my own material. So I hope you watch module 4.2 and 4.3 because we're going to go ahead here with module 4.4, which is the molecular luminescence spectrometry. Okay, and the focus that we will have here is fluorescence. So if you're going to look at this, the overall title or the overall topic that we have, yun yung tinatawag nating luminescence spectroscopy. And here we have fluorescence, phosphorescence, and chemiluminescence. Okay, so if we're going to look at the overall luminescence spectroscopy, ang ano lang natin dito is emission. So at least nakakasulat na ako. Emission of photons from electronically excited states. Okay? And usually, masasabi natin there are two types of luminescence. So ano yung two types nitong luminescence? So one that is a relaxation from singlet excited state, and the other one is relaxation from triplet excited state. So you might ask yourself, ano ba itong mga singlet tsaka triplet state? So what is state that we learned from your Gen Chem? Ano yung state na na-learn nyo sa Gen Chem nyo? Meron tayong tinatawag na ground state, di ba? So when we're talking about ground state, so ano yun? Meron tayong dalawang electrons in an orbital. And if you're going to look at these two electrons, ano yung spin nila? They are usually opposite spin, right? Like this and like that. I think, if I'm not mistaken, that's Pauli's exclusion principle. Okay. Now, what happened here when you go to a singlet state? Mamaya meron tayong diagram na ipapakita dito. Okay. The electron in higher orbital, okay, so the electrons in higher energy orbital has the opposite spin orientation relative, okay, to the electron in the what we call lower orbital. So ang mangyayari nun, let's say ito yung ground state natin. So kapag in-excite natin dito, ang mangyayari ganito. So yan yung tinatawag nating singlet excited state. Now, if we're going to have the triplet state, okay, the excited valence electrons may spontaneously reverse its spin or spin flip. So ang nangyayari instead, pwedeng ganito. So this process na maano natin mamaya, yun yung tinatawag na intersystem crossing. So electron in both orbitals now have the same spin orientation. Okay? So yan yung dalawang type ng luminescence, and how the singlet and the triplet state or singlet or excited state is different from the triplet excited state. So it's just having something to do with the spin orientation. So if we're going to look at some aspect here specifically, so meron tayong tinatawag na molecular fluorescence. So this is a photoluminescence process in which the molecules are excited by absorption of the electromagnetic radiation, and then this relax, giving off excess energy as photons. Okay, so if we're going to look at this fluorescence that we have, it's just a return from the excited singlet state to ground state. Okay, and it doesn't require any spin. This doesn't require change in the orientation spin. So this is a more common relaxation process. Okay, and if you're going to look at this molecular fluorescence, it's more sensitive than absorption. So here it is 1 to 3 orders of magnitude better than absorption. But the problem is its application is limited. Why? Why is molecular fluorescence application is limited compared to absorption? Anyone? Sa 1 dozen student that I have right now? What's the main reason? Anyone want to guess? Bakit sinasabi nating okay, na limited siya? Because it's only applicable to those samples that fluoresce. Or the term that they use is fluorescent sample. Now, how do you know this one we call samples are fluorescent? So if you're going to put a light on it, you will see that there's something that glow. Okay? So that glowing there, or yung tinatawag na fluorescent property there, it's due to the energy being emitted as the electron goes from an excited state going back to the ground state. Okay, so yun yung main principles nitong molecular fluorescence. Okay, and it is measured by exciting the sample at an absorption wavelength. So if you're going to look at this, absorption and excitation is just the same. Okay, so the only difference here is if you have a fluorescent sample, if you excite it, it will yield fluorescent sample. But in absorption, usually ina-absorb niya yung yung energy or light na ina-apply mo. Okay? And if you're going to look at the difference between excitation and emission, usually you excite at a much lower wavelength, and then you will see the emission at longer wavelength. So yun yung tinatawag nating fluorescent wavelength. So example here, the reduced NADH molecule absorbs at 340 and emits photoluminescence at 465. Now question, why do you think the absorption wavelength or the excitation wavelength lower than emission wavelength? Anyone? Ano yung main difference ng dalawa? Let's say in terms of energy. Alin yung mas energetic? Yung 340 or 465? Hello? Okay, so yung 340. And that's the reason why whenever you do fluorescent, you excite your material at higher energy, which means lower or shorter wavelength, and then observe emissions at longer wavelength. Because when you're doing excitation, you are applying energy, you are exciting the electron in your what we call system. So yun yung main reason doon. Okay? And then in addition to that, the way that you measure the emission is at right angle. So hindi to kagaya nung AAS or absorbance where when you put the light here, you're going to observe the reading on this one. So the way that you do it, if you put the light here, you have to observe it 90-degree angle. Okay, so emission is measured at right angles to the incident beam to avoid measuring the incident radiation. And usually there's a short-lived emission that range from 10 to the -5 to 10 to the -8 seconds. That's called fluorescence. Now, if you have a much longer what we call emission, let's say 10 to the -4 seconds or to 10 seconds, ang tawag naman natin doon is phosphorescence. And what's the main difference between phosphorescence and fluorescence? Yes? Ano yung main difference ng dalawa? Okay, yung fluorescence, yan yung sa singlet. Yung phosphorescence, yan yung sa triplet. Remember when we discussed the type of luminescence? So it's just relaxation, okay, from singlet excited state, that's the fluorescence, and relaxation from triplet excited state, and that's what we call phosphorescence. Okay, and if you're going to look at this relaxation process, I think this is the summary that you can have. So dito mo makikita yung difference nun, molecular absorption and fluorescence. Now, molecular absorption here is also equals to what? Yung letter E na dinidiscuss natin. Excitation. Okay? So what the excitation do, depending on a particular wavelength, your electron will be excited from one ground state to an excited state. Now, if your sample is fluorescent, so what happened from time to time, either it undergo this so-called radiative, non-radiative relaxation or fluorescence. Okay, so here in this molecular absorption or your excitation, you could say the light is absorbed. So for dilute sample, you can use the Beers-Lambert law, like what we discussed last week. Okay, ano yung Beer-Lambert law na inaano natin yung formula? A is equals to ABC, di ba? Or we could put it like molecular absorptivity times the path length times the concentration of the thing. Okay, so if we're going to look at the principle behind this molecular observation, which is just similar to excitation, I'm not sure if you encounter this, the Franck-Codon principle. First time nyo ba narinig tong term? Okay. So this Franck-Codon principle just means that the absorption process takes place on a time scale of 10 to the -15 seconds. And this is much faster than what we call the molecular vibration. So ito yung inaano natin, okay, if we have this so-called absorption process. So this is much faster than the molecular vibration, which we're going to talk after this module. Okay. So yung non-radiative relaxation naman, which is this one, and there are two kinds of that. Okay. If we're going to look at this, okay, this is the where the electron is promoted. So bago mangyari ito, kailangan mo na yung excitation. Okay. So the electron is already promoted to a higher vibrational level in the S1 state, okay, the singlet state, when we say S1 state. So you promote it to a higher vibrational level in singlet state than the vibrational level that it was in the ground state. And then what happened there is there's a vibrational deactivation that takes place through intermolecular collision. And if you're going to look at the time scale here, there's around what we call 10 to the -12 second, and that is faster than the fluorescence process. Okay, and I think there's some additional discussion that we have here about the two type of the non-radiative relaxation. So meron tayong tinatawag na vibrational relaxation. Okay, if you're going to look at this, so it's just from one excited state going to lower excited state. So there's a short wavy arrows between the vibrational energy levels, and it takes place during collisions between the excited molecules and molecules of the solvent. Okay, so yan yung tinatawag nating vibrational relaxation. Now, the other form of this non-radiative relaxation, this is just what we term as internal conversion. Okay, and if you're going to look at the internal conversion compared to what we call the vibrational relaxation, it's much what? Longer. Okay, depicted by longer wavy arrows. So relaxation between the lower vibrational levels of an excited electronic state and the higher vibrational level of another what we call electronic state. Okay, so if you're going to look at this internal conversion, this we could say there's a transition between states of the same multiplicity. Multiplicity. Okay, so as you're going to look at this, you could say between states. So this one, the second excited state, okay, going to the first excited state, the first excited state going to the ground state. Okay, so there's we could say a significantly large energy gap between these two. So which do you think here is much larger? This one or this one? Okay, so if you're going to look at this, there's a large energy gap between the two here, and that results to difference in their what we call lifetime. Okay, so yan yung tinatawag nating non-radiative relaxation. And if you're going to look at the thing that we have here, okay, so the first figure we show here, what we call the molecular absorption. Okay, so this is the ground state, the first excited state, and then the second excited state. So this is a hypothetical molecule. So the line in each of these states, yan yung tinatawag nating vibrational state. Okay. Kasi maano kayo, ano yung difference ng mga state na to. Okay, so this is the whole ground state, the first excited state, the second excited state, and each of them has their own what we call vibrational state. So when a band of wavelengths are irradiated, so when you put light or when you introduce energy, so the excited electronic state is what we call populated. Okay, so magkakaroon lang ng what we call excited state when energy is applied. Okay, so either it goes to the first excited state or the second excited state, depending on the energy or light that was irradiated. Now, once the molecules relax, either it goes vibrational relaxation or the so-called fluorescence emission. Okay, so another word that they use for fluorescence is emission. Kasi isa excitation, which is similar to absorption, and then you have what we call the emission. So if you're going to look at this fluorescence emission, okay, it's almost always observed from the lowest lying excited electronic state. Okay, going to this what we call ground state. And it occurs, okay, from the lowest vibrational energy of the first excited state to various vibrational levels of the ground state. Okay, so if you're going to look at the emission, the fluorescence spectrum usually consists of only one band with many closely spaced. So you observe emission usually from the lowest vibrational state known excited state. And it can go maybe to this vibrational state here, this vibrational state, this vibrational state of your ground state. Okay. So the line that terminates the fluorescence bond that is identical in the energy to the line labeled as the wavelength 1. So see this one here? So this is a line with this one. Okay. And then this one is a line with that one, and so on. So all the other emission lines in the fluorescence band are of lower energy, of longer wavelength than the wavelength. So what does this results? Okay, what's the relationship, let's say, of this with this? So what happened here is you're going to have a fluorescence bonds that are mostly lines that are longer in wavelength than the band of the absorbed radiation responsible for their excitation. And you have these so-called stock shifts. Okay, so the shift to longer wavelength is what we call the stock shifts. So when we have these stock shifts, it's just a fluorescence light that is red shifted. So ano ibig sabihin ng red shifted? Longer or shorter?
Longer wavelength.
Okay, so it goes to higher, or what we call longer wavelength. So what happened? Your fluorescence light is red shifted, so longer wavelength than the excitation light relative to the absorbed light. Okay, and there are some process that affect the so-called stock shifts. Isa na dito yung tinatawag na internal conversion. Okay, it can affect your what we call stock shifts. Another way that can what we call affect the stock shifts ay yung tinatawag nating solvent effect. Okay, so if we're going to go further, yung stock shift, it's one field that they utilize in fluorescence to determine the property, yung tinatawag nating fluorophore. So when we say fluorophore, these are fluorescence materials that usually use as what we call a tag. Okay? So ang ginagawa nila, itong mga fluorophore, tinatag nila yung isang system na hindi fluorescence, and they try to monitor ano yung magiging property ng fluorophore once they tag a given system. And one way that they look at this is the so-called stock shifts. Okay? Kasi the stock shifts will give them idea on the nature of the environment of the given system. So one thing that they use is the solvent effect. And solvent effects, usually the excited states reactions can also affect the magnitude of the so-called stock shifts. And if we're going to look, okay, at this what we call excitation and emission wavelength, what can you say here? They are just what? The mirror image of one another. That's what we call like a mirror image rule here. So since the difference between the vibrational states is almost the same for both the ground and the excited state, the absorption spectrum or the excitation spectrum and the fluorescence spectrum, which is the emission spectrum, appear to be the mirror image of one another. Okay? So if we're going to look at this, parang ito yung nangyari dyan eh. So ito yung 1, okay, ito yung 1, ito yung 2, ito yung 2, ito yung 3, ito yung 3, ito yung 4, and this is 4, and that is 5, and that is 5. Okay? So if you're going to look at this, the so-called mirror image, so this is your excitation spectrum, and this is your emission spectrum. Now, your excitation spectrum is usually the same, more or less, with your absorbance. Okay? You only call it excitation if you're collecting it using spectrophlorimeter. But if you just use a UV beam, absorbance yung tawag dyan. So when you have an absorbance spectrum, it reflects, okay, the vibrational levels of the electronically excited state. And then the emission spectrum, what it do, it reflects the vibrational levels, okay, of the electronic ground state. So ito yung tinatawag natin excited state, ito yung ground state. Remember, you get this when you excite the molecules from ground state to excited state. And you get this when your molecules relaxes from the ground state to, from the excited state to ground state. Okay? And this is the mirror image of what we call the spectra that we have for the anthracene. Okay? And the system that you have is anthracene in alcohol. So the excitation spectrum and the emission spectrum. So question so far. Meron ba tayong fluorimeter sa LV? Doon sa naka-experience na makita yung instrument doon. Do we have a spectrophlorimeter in UP Los Baños? Meron ba? Meron ba dati? Parang hindi ko maisip eh. Parang wala ata eh. Hindi ko alam. But I have experience using what we call fluorescent. Okay. I have a paper in a journal called Journal of Fluorescence, and what we did is we looked at the different emission property, absorption and emission properties of a group of antibiotics, okay, at different pH. And if you want to learn more about this, you can go to this link, okay, showing you more about the information that you can gain from fluorescence spectrum. Okay. Now, some of these fluorescent compounds that we usually use is the so-called aromatic compounds. And if you have aromatic compounds, you have what we call the pi system, right? Why do you have a pi system? Dahil dun sa ring. Okay. And the thing with them is you're going to need low energy in its pi to pi star transition. And usually they have this rigid structure. Okay? So merong example dito, all of them are what? Ring system. Yung quinoline, yung indol, saka yung fluorine. And even the so-called polyaromatic hydrocarbons. So are you familiar? What do you call this if I'm going to replace this with C? Anong tawag yan? Anong tawag pag pinalitan ko yung N dito ng C? You call it? Anyone? Ha?
Naphthalene po.
Naphthalene. Okay. And then you can also have it like this. Okay. That's the anthracene. I'm not sure it's anthracene ba to? Because the other one would be, if I'm not mistaken, like this one. And both of them are fluorescent. Okay. So other fluorescent samples are what we call this one. So you have, see the difference here? Yung quinoline mo, dinagdag mo lang ng hydroxyl group, meron ka ng 8 hydroxyl quinoline. And then you have a flavanol, benzoin, and the so-called alizarin. So these are fluorescent chelating agents. Okay, they are, we could say, very useful method. That's why I told you my experience that I have here. I have this fluorophore. I still remember the name because its name is really popular now, yung tinatawag na Alexa. So I have Alexa fluorophore 488. So I use it to tag an antibody, and I try to determine the emission properties. Okay? Because when it's not tagged, hindi fluorescent yung material. So when you tag it, so what happened? Okay, your fluorophore is attached to that biological system, so you can monitor its property. Another way that they use is you collate this time your inorganic species with the fluorescent reagent. And just to give you the idea here, okay, they are really what we call sensitive. So you have an absorption here of 470. So what does this mean? That's your excitation wavelength. So you excite them at that what we call wavelength, and you can see them emitting at this wavelength. So you monitor them, okay? You monitor their signal at this wavelength. And it is a much, we could say, sensitive or more sensitive method compared to the absorbance. Okay? So yan yung, we could say, advantage nitong fluorescence. Now, you can also take advantage yung fluorescence nito, okay, by looking at this so-called fluorescent power emission. So this is just proportional to the radiant power of the excitation beam that is absorbed by the system. And there's a formula for that, but I don't usually do calculations about this. So if you're going to look at the formula here, this is just the power of fluorescence. So we could say that is equals to the quantum efficiency of the fluorescent process times the constant dependent on geometry and other factors times the difference between the power of beam incident of the solution and the power after traversing a length of B. So ito yung pupasok na power dito at yung lalabas na corresponding power. And you can relate it to the Lambert Law. Okay, so to relate the power of the fluorescence to the concentration of the fluorescing species, we write the so-called Lambert Law. Okay, so here we could say the transmitted light over the incident light is just equals to 10 raised to the absorbance, right? Because absorbance is equal negative absorbance, because that is molar absorptivity times B times C. And if we substitute that one, we can do it like this. Now, the other term that they use here is the so-called quantum yield. Okay. So if you're going to look at this, what we call quantum yield, ano lang dyan is like this, F. That's the number of photons that is emitted over the number of photons that was absorbed. So usually in the absence of competing pathways, yung quantum yield mo is equals to what? One. And quantum yield is a very good mode of studying because it's dependent on the biological environment. Okay. Marami pang portion yung molecular sa fluorescence eh. But the one that we cover here are just what we call the steady state. So when we're talking about the steady state, we're just looking at the emission, okay? Because in addition to fluorescence, you can also have this instrument called the lifetime. And the lifetime could provide you more information to the nature of your what we call sample. So the lifetime, that's the time it takes for the electron to go from the lowest vibrational state of the excited state to the ground state. Okay? Ano ba yung lifetime ng fluorescent? Any idea? How long is fluorescent? Because I think if I'm not mistaken, I showed you at the beginning yung difference. So I think fluorescence is what? 10 to the -5 to 10 to the -8 compared to the non-radiative relaxation, which is 10 to the -12. So you can get a lot of information from that, what we call lifetime, but we don't cover here. But in case in the near future, okay, you work in a lab that has that specialty is fluorescence. So there's such thing as a steady state and lifetime state. And both of them is used to what we could determine the thing. Okay? And if we're going to look at the exponential form nitong power of fluorescence, it can be expressed by the Maclaurin series. And it's just like this one. Okay? If 2.303 times the absorbance or molar absorptivity times path length times concentration, you get an absorbance that's less than 0.05. All of the subsequent terms in the bracket in expanded expression can be neglected. Okay? So here, at constant incident light, you could say that the fluorescence is proportional to the concentration. So it's the same principle that we have in absorbance. Okay? The signal is dependent on what we call the concentration. But there is also a limit, just like the Beer's law. Ano yung limit ng Beer's law, if you remember? What's the limit of the Beer's law? Or the Beer's Lambert law? How you recall the limit if it's like this? So ano yung limit niya? High concentration. You also have to be careful with the concentration. Okay? Because what happened, fluorescence is only going to be linear at a certain concentration range. At higher concentration, there are some issues that arises. Okay? So what is this issue? Similar to the Beer's law, hindi na nagiging linear yung relationship nila. What happened at high concentration, there's what we call quenching. How do we define quenching? Anyone? In your own words, ano yung tinatawag na quenching? First time nyo ba narinig yung term na quenching? So yung quenching, if you're going to look at it in terms of fluorescence, that's just any process that decrease the fluorescence intensity of a given substance. Okay? And one reason of this quenching ay yung tinatawag nating higher concentration. And sometimes there are some systems that is dependent on the pressure and the temperature. Okay? So yan yung one, we could say, problem that we usually encounter in fluorescence. Yung tinatawag nating quenching. Okay? So may limit din, kagaya doon sa Beer's Law. Now, if we're going to look at the component of your fluorescence spectrometer, so I think this was just a review of what we have. So the light source that we have, you can have a mercury lamp with this line, the high pressure xenon arc lamp, and the lasers. Okay? I think meron pa akong dadagdagan dito. We have this LED. What's the meaning of LED? Anyone? Anyone?
Light emitting.
It's a light emitting diode. Okay? And just like laser, they have their own wavelength. Okay? Now, I'm telling you, I'm familiar with this because I was able to use some of this during my grad school. Okay? So there's what we call light emitting diode. It's not as powerful as laser. Okay? But it is more powerful, let's say, if you have a xenon arc lamp. The problem with the xenon arc lamp is it's broadband, but the power that you have is parang ano lang yan yung parang ganun. Okay? So at some instances or some wavelength, mataas yung power niya, but not at all wavelength. Unlike yung tinatawag nating lasers and this LED, okay, they are specific. So parang sabi natin na meron siyang specific wavelength that you use. Okay? And if it doesn't match to your what we call target wavelength, so the first thing that the best thing that you're going to do is use a wavelength that is closer to the wavelength that you want to use. Okay? Now, if you're going to use a broadband light source, you need filters or monochromators to select the wavelength that you have. And then you have this PMT, the photomultipliers, and CCD cameras. Okay, I'm familiar with that because I burned one of the CCD cameras of our system when I was new during that time in grad school. Now, the cells that we use, it's always quartz. It's a quartz source. And you have a light tight compartments to minimize stray light. So as much as possible, it's in the dark or i-close mo yung system mo. Because if you have an open system, there could be stray lights that will affect the system. And if you work in the dark, you will see the glowing of your sample when you hit it with your light source. Yun yung maganda dito sa fluorescence na to. Okay, now you can watch the video about the filters and the light source in this link. Okay, so there are terms that usually used for this instrumentation in fluorescence. So sabi nila, if there are wavelength selectors for the excitation and emission spectra are both filters, we call the instrument fluorimeter. Okay, but if both the wavelength selectrodes are monochromators, then we have a spectrofluorimeter. So this is the difference. Kapag monochromator, spectrofluorimeter, kapag meron lang tayong filter, yun yung tinatawag na fluorometer. So sa tingin nyo, alin yung high-tech dito sa dalawa? O alin dito sa tingin nyo yung mas mahal? Practical wise, or should we decide to make your final exam like a true or false question? Okay, so yung mas mahal dito ay yung spectrofluorimeter because monochromator is more expensive than what we call the filter. Okay, what's the simplest filters that we have? Anyone? You can use it every day. Ano yung everyday filter na ginagamit usually natin? Yung shades natin. Okay? So when we use our shades, sometimes it cut off yung shorter wavelength. Okay? Na meron tayo. So that to protect our eye from UV. Okay, that's the simplest filter that you can do. So if you have what we call a fluorometer, so this is what you have. So you have what we call the light source, okay? And then a quartz or what we call a glass lens. So there's an excitation filter here, and there's also an what we call emission filter here. Okay, so filter lang yung meron siya. And I what we call able to use this fluorometer type. Maglalagay lang kami ng glass filter. So what we do is we cut off the light that we pass through here to excite the sample and the light that we collect. Okay, because if you're going to look at the direction of the light, kita nyo to, so it hit the sample there, and then we monitor. It's either an L shape or a T shape. Pwede ka rin magkaroon ng dalawang ano may ano pa rin dito emission detector. It's also possible like a T-shaped thing. Okay. Now, when you have a fluorimeter, it's simple, it's rugged, it's low cost, it's compact. Emission is measured at right angles to the mercury arc lamp source. And the fluorescence radiation is emitted in all directions, and the 90-degree geometry avoids the detector viewing the source. Okay. And if you have a spectrofluorimeter like this one, like Perkin-Elmer, okay, ganito yung system mo. So you have the light source here. There's a monochromator there. Okay, so it passed through the system here, and then you have the sample cell. So as you can see, it's also L-shaped for 90 degrees Celsius. So it passed through another monochromator here until it reaches your transducer or your detector, and then the readout system. Okay. So you have two grating monochromators for both excitation and emission. So you view emission at right angle also, and this is more useful in quantitative analysis. Okay. Now, you can watch more on the video about the molecular fluorescence here. Okay, and they contain more, we could say, information about molecular fluorescence. And then I'm going to still have another what we call material after this material from IC. Yung ginagamit kong material dito. Now, let's go with the molecular phosphorescence. We could say, to tell you in advance, I don't have any experience of this phosphorescence. Okay, but if you're going to look at the principle, it's the same as fluorescence. And the only difference that I can tell you about them is what? Fluorescence is relaxation from singlet excited state Y, while phosphorescent is relaxation from triplet relaxation state. Okay, so if there's needs to consider if the electronic state of the molecule, so if you have a ground singlet state, you can have a molecular electronic state where all electron spins are paired and is at ground state. You can have a doublet state, so the ground state of a free radical. You can have an excited singlet state where one of the pair of electrons in the molecule is excited to a higher energy level, but they have opposite spin. Okay, and the triplet states, that's the one where their spins become paired or parallel. So yan yung tawag natin yun sa phosphorescence. So remember here, okay? So this is the singlet excited state, and this is the triplet excited state. So phosphorescence happen, okay, when an excited triplet state relaxes to a ground singlet state compared to the molecular fluorescence, which you have a singlet state that relaxes to the ground state. Now, the transition is phosphorescence is very slow compared with, say, with fluorescence. So usually here, the absorbed radiation is re-emitted at a lower intensity for up to several hours after excitation. So I think that's what makes it not so popular. Okay? Because it is a longer lifetime. So hindi rapid yung analysis. But there are examples of this phosphorescence material. So maybe you're familiar with the glow in the dark toys, yung sticker, yung paint, yung wristwatch, or yung tinatawag na clock dials. Okay? You're familiar with this one, right? We have a glow stick. They last for a while. Unlike sa fluorescence, they only last when you hit them. And then after when the light source is gone, you don't see them anymore. Okay? So most of the phosphorescent material, they glow after being charged with bright light. And the glow slowly fades out sometime in a few hours in the dark room. I don't know if you're familiar yung parang mga planets or yung stars that you put in your roof, yung sa room nyo, sa ceiling. Ceiling ba tawag? Basta pag pinatay nyo yung ilaw, makikita nyo mag-glow. I don't know if you have it when you're a kid. Because I have it with my son before when she was still a kid. So when we turn off the light, you could see some of the glowing shapes. Okay? Para hindi naman masyadong madilim doon sa kwarto, sa room niya. So that is what we call fluorescence. And you can look more about this. No, that's not fluorescence, that's phosphorescence. You can know more about phosphorescence if you go to this what we call link. Okay? Supplemental information. Now, the next thing that we're going to do is this so-called chemiluminescence. So, if you're going to look at the word, the one that we discussed so far is just luminescence. And we could say this is just what? At the beginning, ano yung sabi natin sa luminescence? It's just the emission of photons from electronically excited states. But when we're talking about chemiluminescence, what's the difference of this? Yung letter na yan. Okay? So this happened from chemical reaction. So it involves chemical reactions that yield electronically excited species that emit light as they return to the ground state. Okay? So, ano yung pagkakaiba nito doon sa luminescence? What is the main difference of chemiluminescence from luminescence? So here, you need a chemical reaction, and sometimes most of these are what? Biological in nature. Diba? Yung firefly, anong tawag sa atin sa firefly? Yung may alitap-tap? Is that what firefly is?
Yes, po.
So, sorry, hindi ko na kasi nagagamit. That's why I'm just trying to make sure. But you see them, right? When you're a kid, there's something, the insects that just light up. Since they are biological system, ang tawag natin doon is bioluminescence. Okay? And the examples of this bioluminescence ay yung tinatawag natin nangyayari sa firefly, sa jellyfish, at saka doon sa crustacean. And I have studied about this so-called, I'm not sure. Are you familiar with holoturia? I studied yung tinatawag naming fluorescent holoturians. But I think the way that we look at it is due to bioluminescence. So these are what? I'm not sure if you're familiar with sea cucumbers. And if I'm not mistaken, I presented a poster about it on the KKP, one of the annual meeting of the KKP. Okay. So what we do there, we excite some of these sample that we have, and we determine how it can be used like chloroform. Okay. So when you're doing chemiluminescence, it's simple instrumentation that is required because there's no external radiation is needed for excitation. Usually saan? Yung system mismo ang nagpro-produce ng light. So wala kang tinatawag na light source. Okay? Instrument is consists only of a reaction vessel and a PMT, photomultiplier tube. So all you need to do is what? seal the system in a container or reaction vessel, and then make sure na meron kang detector na naka-allotted doon. Yung detector na yun, yung photomultiplier tube. There's no wavelength selection required because the only source of radiation is the emission caused by the chemical reaction. So, hindi mo kailangan ng light source. Because yung system mismo, yun na yung mag-produce ng light. Okay? So, all you need to do is what? Monitor yung na-produce na luminescence doon. Okay? So, it's a highly sensitive process. The detection limit can be ranged from ppm to ppb or lower. Okay? And then the application would include the determination of gases such as oxides of nitrogen, ozone, and sulfur. You can also use it to determine inorganic species or the so-called immunoassay techniques. So if you're going to look at the reaction that you have here, these are just some. So we have, you have NO and ozone. You produce NO2, that is, we could say, excited. And what happened there? That can generate some light. It produces some chemiluminescence reaction. Or you have hydrogen with sulfur oxide. It can produce the sulfur 2 star, and that can also generate you some light. Okay, and I don't know if you watch CSI or any forensic science stuff. You can also use it in the detection of the blood in crime scene. So I don't know if you see it. Sometimes they have like a flashlight with some filter, and then they turn up the light, they light it up, and you see some stuff that glow. Okay? And I think in last week's episode, okay, yung hindi tayo nagkaklase, they talk about luminol. So they sprayed it across an area and it reacts with the iron in the hemoglobin, and it emits a blue glow that can be seen in a darkened room. Okay, because the CSI here starts from 10 to 11 and our class is around 10:30. Okay, so there's a CSI Vegas that you have there. But if you're going to look at here, they just turn off the light, they spray that luminol, and if there's some blood on it, okay, it will give you a blue glow. So that's the principle that you have here. So if you're going to look at the chemical reaction that happened there, ito yun nangyayari. So maybe this is in preparation for you if you end up in what we call the forensics. Unfortunately, walang forensic science dyan, di ba? Do we have a forensic science program there? In the Philippines? May course na? Okay. Because I tell you, I have a lot of batchmates who are in crime lab at this time. Okay, the provincial commander in crime lab in Cavite, that's a batchmate of mine. Okay, and one of the officer there, that's also a batchmate of mine, and a nephew of Mamigo. Here in the US, the school that I have, we have forensic science program, and there are more forensic science majors compared to chemistry and biochemistry. And we suspect that they went to the forensic science program because of the CSI. And most of them usually don't last in 2 years because after they take Orgo, they say bye-bye. They cannot survive the Orgo, just like you, right? Tama ba? 43 pa rin yung pangsala sa inyo? Is it still 43, 44 for the Orgo courses? Yeah, the 40, yun pa rin. The 43 and the 44 will decide whether you stay in chemistry and agricultural chemistry program or you need to shift to another course. Okay? And I'm proud to say, take one ako sa courses na yun. Okay? with Dr. Peralta and Dr. Hernandez. So if you're going to look at the chemiluminescence that happens here, so in the presence of iron, okay, your hydrogen peroxide decomposes to form oxygen. And then the diion reacts with oxygen, producing the unstable organic peroxide intermediate. And this intermediate decomposes from higher energy states to the ground state, emitting a quantum. So this is just, we could say, the chemistry of luminol. How luminol was able, okay, to determine the presence of blood in crime scene. But this can only happen, okay, if it's in the dark, okay? Because what happened there, when you add luminol, it can produce the so-called diion intermediate. And that's the one that's produced or that emits the photon. Okay? So when you have an HB there, that means it's a light. So if it's in the product, that means it's produced. If it's in the reactant, that means it's used. So that's the difference of this HB, whether you are in the reactant side or the product side. Okay? So these are the more pages of this chemiluminescence, and I think we end this thing here. Wala pa rin akong ibibigay na quiz. I will announce it after we meet on Thursday. But in addition to that, as I have told you, I have another material on molecular spectroscopy. I have not sent it to you yet, but I'm going to what we call maybe send this to you, or we could just say look at the video that we have here. This is just a specific example that we have. So it's still specifically on fluorescence spectroscopy. Okay? So ito yung summary ng lahat. Anyone know what they call this, what we call reactions that is happening? Are you familiar with this? Jablonski diagram. Is it the first time for you to hear that term? It names after a guy named Jablonski. Okay. So if this is the first time that you hear it, at least you learn something today about what we call this Jablonski diagram. Okay. So if you're going to look at this Jablonski diagram, this is just a diagram that illustrates the electronic states of a molecule and the transitions between them. Okay. And this, the name of Jablonski is what? Alexander Jablonski. So you see there an ski, just like Jaworski. So yung ethnicity niya is more on Polish. Okay. So if you're going to look at this Jablonski diagram that is shown here, so it shows you the different processes that happen. And as you could see here, it's just the same as what you have. I have what we call discussed earlier. So you have here the absorption or excitation. It's around 10 to the negative 15 seconds. Okay. And then what happened? You can have this internal conversion, okay, or vibrational relaxation. So ano yung difference kapag between what we call states, yun yung internal conversion. So kapag between vibrational energy states here, maybe yung dito lang or doon sa side na yon, yun yung tinatawag na vibrational relaxation. Okay? Now, dito natin makikita saan yung singlet state at yung tinatawag na triplet state. Because in our original thing that we have there, we just put this as what? E0, right? And then E1. and then E2. Now we're going to make it specific. That's the reason I said I think I need additional slides for this part. So yung S dyan, ibig sabihin yan yung singlet state. Now, if you're going to look at the singlet state, see the red one here? So it's the lowest vibrational state in the excited state or in the singlet state specifically going to the ground state. Yan yung fluorescence, which is 10 to the 9 to 10 to the negative 7. Okay? Now, meron pang additional process dito that I think we didn't discuss doon sa material natin. So, meron tayong tinatawag na intersystem crossing. Okay? So, what is this intersystem crossing? Ito yun. So, when we have this intersystem crossing, it refers to non-radiative transition between states of different multiplicity. Can we say they are of different multiplicity? Yes, because this is a singlet and this is a triplet. Okay? And then you can also have here this so-called non-radiative relaxation, but this one coming from the triplet state. So dito mo makikita talaga yung difference nung tinatawag nating fluorescence saka phosphorescence. Now, you can have this so-called delayed fluorescence. Okay? And then we have also what we call the quenching and the other non-radiative relaxation. So this gives you, the Jablonski diagrams gives you more information about what's really happening, okay, in the process of luminescence. So I hope this gives you more detail, okay, about this luminescence thing. Questions so far? Okay, so if we have this fluorescence, so if you're going to look at this, this is just from what? The lowest vibrational state of the excited state or singlet state going to the ground state. Okay, so phosphorescence, it passes through the triplet state and going to the ground state. And the intersystem crossing is the what we call non-radiative transition between the singlet state and the triplet state. And if you're going to show here, I'm going to show you the so-called mirror image thing at yung tinatawag nating stock shifts. So this is an excitation and emission profile. So the excitation is just similar to the absorption that we discussed. Now, the emission is just the mirror image of that. Now, sometimes the stock shift is affected. How is it going to be affected? It can become wider or narrower, depending on the system. Okay? Now, the experimental setup, which I already discussed to you, as you could see, there's always a 90-degree angle. So this is the light source. There's an excitation monochromator. You pass it through your sample and you determine, so from the incident light to the transmitted light, so you determine the emission at 90-degree angle. It passed through a monochromator. So the emission spectrum, you can have what we call two spectrum here. This excitation spectrum and emission spectrum. And they're both useful. Okay. But usually the one that we usually use if you want to do fluorescence is just the emission. Now, what can you say when I say whenever you do emission, what we call analysis, you usually get to know the absorbance. Why? Why is it that you need to determine the absorbance first of the system before you do fluorescence? Anyone? Maybe you don't know the answer, but maybe some of you can guess. Anong use ng absorbance before you go emission? Before you do emission? Anyone wants to guess? So what do you see here? What is this? What do you call that highest peak? That's what we call the lambda max. Ano tawag yan? Yung sumagot?
Lambda max.
Okay, so that's the lambda max. So what's the use of the lambda max? Usually, once you get the lambda max in your absorbance, you can use that also as your lambda excitation. Okay, and the rule that they have is if you want to get the emission, make sure the absorbance that you have will not go over 0.1. Kasi pag mas mataas na, baka masyadong malakas yung emission niya. So they try to maintain it around 0.1, yung lambda excitation. So that's the use of the absorbance. It will tell you what's the lambda max that you're going to use. What's the wavelength that you use to excite your sample. Okay? Now, ano yung nature ng study na ginagawa dito sa fluorescence spectra? This could be a BS thesis. Okay? So usually they look at the effect of the solvent and the environment. And they can look at the difference on the fluorescence spectra that they can get. The solvent polarity and viscosity. So anong ibig sabihin nito? If you are in a polar solvent, maybe the emission spectra or the lambda M, okay, the maximum emission is different or changing. How? Well, it depends on what? Saan nakaka-affect yung polarity? Anyone? Anong process sa fluorescence na pwedeng maapektuhan due to solvent polarity and viscosity? What's happening during the fluorescence spectra? There is excitation and emission. So sa tingin nyo, alin yung maapektuhan? Due to the solvent polarity and viscosity. Usually, it can be the excitation or it can be the emission. Okay? But if you're just looking at the steady state, mostly it's the excitation. Why? Well, depending on the polarity of your sample, it's going to behave differently at different polarity. Pag viscous yung sample mo, anong mangyayari? Hindi siya agad ma-excite, di ba? Kasi maapektuhan siya ng environment. So you can also have the rate of solvent relaxation. Okay? You can probe conformational changes. You can determine the rigidity of the local environment. You can also look at the internal charge transfer. You can also look at the proton transfer and the excitation state reaction. Or you can look at the probe-probe interaction, the one that I told you. You tag it. Okay? So this is really very useful. Sabi ko nga, pang BS thesis to, if someone is doing it in UP Los Baños. Okay, now, how does the solvent affect it? So here, makikita nyo. Okay? So typically, the fluorophore, they have a larger dipole moment in the excited state than the ground state. So the solvent would shift the emission to lower energy due to the stabilization of the excited state by the polar solvent molecule. So naapektuhan kapag nasa excited state na siya. So what happened? The solvent polarity increases, the effect becomes larger. So the less polar solvent kita nyo dito, more polar solvent kita nyo, mas mababa. And how it affects your emission spectra? There. So meron itong tinatawag nating prodan. So this is the structure. And as you could see, there are the aromatic ring. Okay? So the increase of solvent polarity leads to larger redshift. So ano yun nandito? Water o heptane? Saan yung water? Saan nag-i-increase yung water? Dito, let's say A, o dito? B. Ano sagot nyo? Nasaan mas maraming water? Sa A or sa B? Okay? So usually it's in the A. And you know, it's really what we call a very simple what we call experiment. Ang ginagawa nila, okay, they just vary the composition of a polar and a non-polar solvent. So yan yung na-observe nila sa product. Okay? Now, the example that I can give you, ito yung tinatawag nating fluorescent. So what do you think is the difference between the two here? Pag sinabi ko, alin dito yung high pH, alin dito yung low pH? Let's say A or B. So ano yung A, ano yung B in terms of pH? Saan yung high pH, saan yung low pH? You should be able to tell based on the structure, right? Sige nga. Ano yung A? Is it low pH or high pH? Tingnan nga natin. Ano yung A? Ano ba yung high pH? Basic, acidic? Ano yan? Basic or acidic? Basic. So, ano ba pwede natin sabihin? A is to acidic, B is to basic? Why? Bakit ko sinabing acidic to? Ano yung evidence? Just trying to see kung gaano nyo naintindihan yung basic chemistry nyo. Why do you think I say this is an acidic pH? It's something to do with this one, right? Acidic pH, which means protonated. At high pH or basic pH, you see, wala yung H. So, which do you think is more fluorescence here? The A or the B? Which is fluorescent here? Wala lang. Because this is what we call a structure wherein, based on the change in pH, your materials become more fluorescent. So the B is the one that is more, we could say, fluorescent. And if you're going to look at the emission and excitation spectra of this is what you have. And this is what? A mirror image. Okay? And just to show you proof that indeed the basic pH is the higher or the more fluorescence, look at the intensity. So yung higher pH mo, mas mataas yung intensity niya. And if you're going to put salt on it, look what happened. So this is from one that contains 0 molar of NaI going to 12.2 molar of NaI. Ano masasabi nyo? Kapag merong salt na present doon, mas mababa yung kanyang emission. So naapektuhan yung pag-excite niya. So kung hindi masyado na-excite, wala kang mai-emit. Why? Because of the presence of the other ions in a given system. So parang meron silang tinatawag na crowding. Okay? So if you're going to look at the exact example of this fluorescent, this is how it looks like in a solid form. But if you dissolve it and excite it with a light around 400 something, if you're going to look here, so it's around 480, so it will excite something, emit something at 520. So this is one of the experiments that we do here. Okay, the fluorescent sample, fluorescent. So question. So at least hindi na sayang yung time natin. Okay. So tanong. So wala tayong spectrofluorimeter sa IC. This is a very good research thing. And the way I do my research here, meron kaming absorbance, tapos i-relate namin yung sa emission. And sometimes we put it in the IR. And if it's a biological system, we put it in the CD. So in my other lab, in your college, whenever I work there are 4 instruments that are running at the same time. And what I do, from one instrument, I just transfer the cuvette to another instrument to get the other reading. So yun yung tinatawag nating multitasking. And unfortunately, most of our students at this time, pwede lang silang mag-multitask when they're using their cell phone. So that's one of the traits that we miss, okay, from the old school that we have. Yun yung nakikita namin doon sa mga bagong students na napunta dito sa US. Okay. So question. So I'll turn off the recording. Question.